Good day, everyone. We are Group 5 and we will be discussing the first part of our laboratory activities on systematic zoology. Firstly, it is important to know about nomenclature. Nomenclature is a system of naming organisms consisting of rules that would allow each organism to be identified in a unique way. Linnaean nomenclature is known for the binomial system, where organisms are named with two Latin words consisting of genus and species. One example of this is the scientific name of humans, known as the Homo sapien, where the word Homo is the genus and the word sapiens would be the species. The Linnaean nomenclature was found by Carolus Linnaeus. Carolus Linnaeus was a Swedish botanist, zoologist, taxonomist, and physician who introduced the Linnaean nomenclature. He is also considered to be the father of modern taxonomy. Carolus Linnaeus was also the one who found the system of classification called the Linnaean hierarchy, consisting of kingdom, phylum, class, order, family, genus, and species, with species being the most specific in the classification. When naming organisms, there are four types of methods used to name new discoveries. First would be descriptive, which names an organism after its physical characteristics. An example would be the Staphylococcus aureus, which describes the organism to have grape-like clusters of spears and to be golden in color. Next would be the scientist's name, which names the organism after the person or scientist who discovered it. An example would be Listeria, which is an organism found by Joseph Lister. Next is geographic places, which names an organism based on the location where it was found. An example would be the Blastomyces brasiliensis, which is an organism found in Brazil. And the last would be organizations, which names organisms based on organizations they're related to in some way. An example would be Legionella, which is an organism related to the American Legion. This activity is separated into three groups: the animal-like protists, the phylum Porifera, and the phylum Cnidaria.
Animal-like protists. Objectives: to identify the protozoan representatives and describe the structure of the organelles and their functions. Materials: prepared slides are Euglena, Trypanosoma, Amoeba, Foraminifera, Plasmodium, Paramecium, and Vorticella. The used microscope is compound light microscope. The animal-like protists are commonly known as protozoans. They are believed to be descendants from the form of life that gave rise to multicellular organisms. This group includes unicellular organisms, and it is diverse in modes of nutrition and modes of existence. Some are capable of locomotion by pseudopodia, flagellia, or cilia. Its reproduction is asexual by fission, budding, or cyst, and sexual by ligation. Systematic classification. Phylum Amoebozoa. Most are unicellular and most produce lobate pseudopodia. They are free-living heterophs that feed by engulfing the other cells with their pseudopodia. This genus is amoeba, an inhabitant of slow streams and ponds. Its cytoplasm has two regions consisting of ectoplasm and endoplasm. It's suspended in the endoplasm are nucleus, contractile vacuole, and food vacuole. As visible in the image is lobopodia. Next is phylum Euglenida. Its shape of cell maintained by pellicle, formed by interlocking strips of protein beneath the cell membrane that are associated with linked microtubules, arranged in regular pattern, also beneath the cell membrane. Some species secrete a mucus chlorica, mostly marine, although much of the described species are freshwater. Its genus is Euglena. Common name is algae. Found in freshwater streams and ponds, and its organism is spindle-shaped, pointed at the posterior end and blunt at the anterior end. At the center of the body is an oval nucleus as seen on the image. Next is phylum Kinetoplastida. They possess a single large, elongate mitochondrion with disodial crysting and conspicuous disodial concentration of mDNA. Mostly are primarily free-living heterotrophs in marine and freshwater environments. Its genus is Trypanosoma. It spends most of their life in the blood and outer body fluids of vertebrate hosts, transmitted by tsetse fly and best observed in blood smear of an infected individual. Its body is spindle-shaped with posterior and somewhat pointed. It is single and central nucleus is present. Next is phylum Granuloreticulosa. Its cell is surrounded by plasma membrane which may be supported by organic agglutinated or calcareous test. Locomotion and feeding involves long thin pseudopodia called reticulopodia, which branch and fuse to form a network. They are ubiquitous in all aquatic habitats and all depths. Its genus is Foraminifera and common name being forams. These shells are sarcoids, lives in oceans with few in fresh and brackish water, usually have many chambers and are made of calcium carbonate, and it is slender in pseudopodia is visible. Next is phylum Apicoplexa. All apicomplexans are parasitic, and the group is characterized by the presence of a unique combination of organelles at the anterior end of the cell called the apical complex. Its genus is Plasmodium, and the common name is malaria parasite, causative agent of an infectious disease, malaria. It spends a large part of its life cycle within the host's red blood cells and reproduces asexually via schizogony and sexually via fusion of gametes. RBCs infected with schizons can be seen with ring and appearance over of dark spots. Next is phylum Cilicia. They derive their name from the Latin word for eyelash, cilia. Ciliates are very common in benthic and planktonic communities in marine, brackish, and freshwater habitats, as well as damp soils. Its genus is Paramecium, and common name is sleeper animalcule. Abundant in ponds or sluggish streams with aquatic plants and decaying organic matter, and it is slipper-shaped and the entire body is covered with cilia. Within the cytoplasm is a prominent large macronucleus, and beside it is a micronucleus, as seen on the image. Following is Vorticella. It is common in fresh water and shaped like an inverted blood and attached by a contractile stalk. Cilia are confined in the oral region and has a bean-shaped macronucleus and a dot-like micronucleus.
The next part of our discussion the next part of our discussion would be the phylum Porifera. The phylum Porifera are comprised of the sponges. They are considered to be the lowest forms of multicellular animals because neither tooth tissues nor organs are present. Sponges are exclusively aquatic organisms, and they are usually sessile or stationary. The taxonomy of the sponges is mostly based on the details of their endoskeleton. They can reproduce asexually by budding, fragmentation, regeneration, and gemule formation, and sexually by fertilization forming free-swimming ciliated larvae. The objectives of this part of the activity is to identify the parts and organization of sponges, and to differentiate representative species of sponges from each other. The materials that we would require would be a compound light microscope, a hand lens, prepared slides of Leucosolenia spicules, and Grantia. Preserved specimens of Euplecteria, Spongia, and Carteria spongia would also be used. Systematic classifications. The class Calcarea. The members of this class have spicules made up of calcium carbonate. They are needle-shaped, 3 or 4 rayed. These sponges also exhibit 3 types of canal systems: the asconoid, syconoid, and leuconoid. All members of this class are also marine dwellers. The genus Leucosolenia of class Calcarea is a simple sponge growing abundantly on the rocks near the seashores just below the low tide mark. It has an opening at the summit known as the osculum. It has an asconoid type of canal system. The genus Grantia of class Calcarea is a common solitary calcareous sponge, gray or light brown in color, inhabiting the salt water along the coast near the low tide mark. It has a large central cavity called the spongiocele. It also has incurrent and radial canals that lie alternately and readily around the spongiocele. The class Hexactinellida. Members of this class are so-called glass sponges with siliceous six-rayed spicules. They have flagellated radial canals or chambers in simple syconoid or leuconoid arrangement. All are marine species and most are deep-sea dwellers. The genus Euplectella of class Hexactinellida is especially abundant at the bottom of deep-sea waters. It has a long curved cylindrical body containing a large spongiocele. It also has an opening at its extremities known as the osculum. Class Demospongii. The skeleton of these sponges is made up of either siliceous spicules that are not six-rayed or spongin fibers, or a combination of both. They exhibit the leuconoid type of canal system. Only one family is found in freshwater and all the others are marine dwellers. The genus Spongia of class Demospongii is popularly known as the common commercial bat sponge. It is globose or hemispherical in shape, and it is colonial and brown in color. Large openings called the oscula are found throughout its surface. Its skeleton consists of a complex network of spongin fibers arranged in anastomosing pattern. The genus Carteriaspongia of class Demospongiae has a broad leathery plate body. It is attached to the substratum by means of a root-like stalk, and it is commonly found in shallow marine water.
Now, as for our last phylum is the phylum Cnidaria. So the phylum Cnidaria are radial in symmetry, meaning similar parts are arranged symmetrically around a central axis. It has a presence of stinging tentacles and a simple gut with only one opening, also known as a gastrovascular cavity. Considered to be the most primitive of the neumatozoans, meaning they are true multicellular animals. They exhibit a tissue level of organization. They also have an exoskeleton or endoskeleton made of chitinous, calcareous, or protein components. They are the only group of animals with nematocysts for defense and capturing prey, but it can also be used for locomotion. Nematocysts are large organelles produced from the Golgi apparatus as a secretory product within a specialized cell, the nematocyte or cnidocyte. Cnidarians have two body forms. the polyp and medusa, which often occur within the life cycle of a single cnidarian. They are the most primitive of animals whose cells are organized into distinct tissues, but they also lack organs. The phylum Cnidaria is made up of four classes: the Hydrozoa or Hydrozoans, the Scyphozoa, the Scyphozoans, the Anthozoa, the Anthozoans, and the Cubozoa, the Cubozoans. The objectives include identifying the basic parts and organization of cnidarians and differentiating the representative species of cnidarians from each other. The materials required for this part of the exercise is a compound light microscope, a prepared slides of Hydra and Obelia, and preserved specimens of Metridium, Aurelia, Acropora, Fungia, Tubipora, and Sacrophyton. Now let's move on to the systemic classifications of the phylum Cnidaria. In the class Hydrozoa, we have the member of this class are either solitary or colonial. They are also relatively small and inconspicuous and inhabits either freshwater or marine water. They exhibit both polyp and medusa stages in their life cycle. Their mesoglea are noncellular and their gonads are epidermal. They also have cells that are used for stinging called cnidocytes. Now, under the Hydrozoa is the Hydra, or commonly known as the Hydra. These have these species have no circulatory systems, heart, or blood vessels. Their bodies are only two cell layers thick, so oxygen diffuses into areas of the cells that are low in oxygen. They have no skin, but the outer layer of cells protects them. They also do not have a stomach, but they do have an inner layer of cells that secretes enzymes that digest the food. We can see here the bud, mouth opening, pedal disc, and tentacles, which were the parts that we were required to identify in our worksheet exercises. Now we have another class hydrozoan, also which is the Obelia, also known as the sea fur. It is often brilliantly colored animals that exhibit radial symmetry. It is an erect, bushy colonial hydroid that inhabits the sea and is usually found attached to shells, rocks, and algae. The polyp and medusa stages are both present in their life cycle. The polyp form can be seen by the naked eye, but the medusa form is microscopic. We can see here from the image the medusa bud, hypostome, and hydrocolus. Now for the class Cyphozoa, they are considered as the true jellyfish found inhabiting coastal marine waters, while some are deep sea dwellers. They are also solitary, and the defining characteristic of this class is that the medusa is the prominent stage in the life cycle, although there is a polyp stage present. Their mesoglea is thicker and cellular, and their gonads are gastrodermal. Under the class Cyphozoan, we have the genus Aurelia, also known as moon jelly. They are solitary, and as you can see, they are the true jellyfishes that you see on the sea. They are also known as Scyphotho medusa. They are common cosmopolitan species that inhabits temperate and tropical sea waters. They have semi-transparent umbrella, more discoidal, and less cute. Now we have another class called the class Actiniaria. It contains the true sea anemones. These are solitary hexacoralia, which never possess a calcareous skeleton. Their base are rounded or forming an adherent disc with or without basilar muscles. Now, under the class Actiniaria is the genus Metridium, are also known as plumose anemone. They are characterized by their numerous thread-like tentacles extending from atop a smooth cylindrical column and can vary from a few centimeters in height up to 1 meter or more. In larger specimens, the oral disc becomes densely curved and frilly. Now for the class Anthozoa, they are either solitary or colonial polyps, and their medusa stage is completely absent. The anthozoan polyp is larger and complex than the polyps of other classes. Their mesoglea is highly cellular and well developed, and their gonads are endodermal. They are also mostly marine species and predominantly sessile or stationary animals. So under the class Anthozoa, we have the genus Acropora, or commonly known as the staghorn coral. All Acropora corals end with a large axial corallite. It is a key feature that distinguishes the Acropora from other corals. It is located right on the tip of each branch, as you can see from the labels on the image on the right. It is the most abundant corals of most reefs of Indo-Pacific. They could also be colored as pale, cream, brown, or dark brown. Another under the class Anthozoa is the genus Fungia, or commonly known as mushroom coral. They are usually solitary and generally free-living except for the juveniles. Their polyps are approximately circular and up to 280 milliliters in diameter. Their septal teeth are triangular, pointed, and usually have well-defined central ribs. Their tentacular lobes may be present. The costal spines are tall, smooth, and conical. We can see from the image its central mouth and the septa surrounding the mouth. Another under the class Anthozoa is the Tubipora, commonly known as the organ pipe coral. It, they occur on reefs in shallow waters of the Indian and Pacific Oceans and is characterized by long, parallel, upright polyps or stalks supported by a skeleton of rigid tubes of calcium carbonate. The tentacles of the polyps are sometimes green and the skeleton is bright red or crimson. After the soft parts die, the skeleton remains as a brightly colored mass of tubes that resemble organ pipes, hence the common name. So the last genus so that's all for the presentation. Thank you for listening. Hope you guys learned a lot and have a nice day. Thank you.